What you are seeing here is the absolute purity standard required for purified water and water for injection in pharmaceutical manufacturing. For decades, engineers managed the branching fluid paths in these water systems using the 6D rule. This dictated that a dead leg, a closed branch pipe where water sits idle, could not be longer than 6 times the diameter of the pipe itself. As microbiological standards tightened, the industry adopted the stricter 3D rule to further minimize the volume of idle water. Yet, modern facilities strictly adhering to this 3D rule still experience severe, batch-ruining endotoxin spikes in their distribution loops. Traditional engineering operates on a baseline assumption that limiting the physical length of a dead leg is an adequate way to maintain water quality. The math and the biology of modern cGMP environments prove that mitigating dead leg length is insufficient. To guarantee purity, systems must achieve continuous fluid shear stress. Here, a traditional T-junction on the left contrasts with a flush zero dead leg or ZDL block valve on the right. The FDA increasingly mandates this continuous shear architecture, pushing away from legacy 3D rule geometry. Achieving regulatory compliance requires engineering a geometric environment where fluid stagnation cannot physically exist. To understand why the 3D rule fails, we have to look at the fluid dynamics inside a standard T-valve branch. This cross-section shows the main water line with a closed branch, inherently removing fluid from the main loop's primary shear stress. As fluid enters that closed branch, its kinetic energy drops, creating a distinct low-velocity zone isolated from the main flow. This low-velocity zone is exactly where biological threats like Pseudomonas bacteria thrive. Without adequate shear stress to sweep the walls, bacteria adhere directly to the stainless steel surface, secreting a protective matrix to form a solid biofilm that shields the colony. The trigger for a contamination event is often a simple mechanical shift. When fluid dynamics change or system pressure fluctuates, the physical stress on the pipe wall shifts. That shift shears off fragments of the biofilm, releasing high concentrations of endotoxins directly into the system. A single localized biofilm in one minor branch valve initiates a contamination cascade, circulating pyrogens throughout the entire distribution loop and compromising multi-million-dollar batches. As long as a geometric branch exists, a biological breeding ground exists. Compliance with the 3D rule offers a false sense of security. The engineering solution is to eliminate the branch pipe entirely using a zero dead leg diaphragm valve, also known as a block valve. This 3D wireframe illustrates the geometric shift of ZDL architecture. Instead of placing the valve seat at the end of a perpendicular pipe, the weir is machined directly into the main distribution line. When closed, the flexible diaphragm sits perfectly flush with the primary flow interface. The immediate volumetric result is that there is zero physical space for fluid to pool. This geometry dictates a state of continuous turbulence. The flow from the main pipe violently and continuously washes directly over the flush diaphragm and weir. The shear force of the main loop's velocity perpetually and actively sweeps the entire valve surface. There are no isolated pockets, and there are no stagnant boundary layers where velocity drops. This continuous sweeping physically prevents bacterial adhesion from initiating. The bacteria can never establish a foothold on the steel. ZDL architecture achieves absolute purity by utilizing the system's own kinetic energy to engineer an environment actively hostile to stagnation. This continuous flow geometry also dictates the success or failure of routine facility maintenance, specifically clean-in-place and steam-in-place cycles. This data table compares the sterilization efficacy of standard 3D T-valves against ZDL geometry. Standard dead legs are notoriously difficult to validate. When steam flows into a traditional T-valve, it fails to reach the very end of the closed branch, leaving an unsterilized void space. The lack of flow velocity inside the dead leg prevents the cleaning agents or high-temperature steam from fully penetrating the branch apex. By contrast, in a ZDL configuration, the vapor sweeps aggressively across the flush weir and diaphragm. The fully swept design ensures 100% surface contact with the sterilizing agent. This total thermal and chemical contact drops the risk of biofilm survival to near absolute zero, meeting the rigorous demands of modern ASME BPE standards. A piping architecture is only as safe as its hardest-to-clean component. ZDL geometry is the physical prerequisite for a truly validated sterilization cycle. Zooming out from the micromechanics of a single valve reveals the macro stakes of operating a high-purity pharmaceutical water system. The FDA and the International Society for Pharmaceutical Engineering evaluate these systems through a regulatory framework called Quality by Design. Quality by Design requires building safety and compliance directly into the physical mechanics of the equipment, rather than relying on testing for quality after the process is complete. Integrating ZDL architecture drastically simplifies a facility's installation and operational qualification validation process. By structurally eliminating stagnant zones, engineers remove the exact high-risk sampling geometries that typically trigger audit failures. The financial stakes are absolute. A single contamination event traced to a dead leg will trigger an FDA 483 warning letter and force the immediate scrapping of multi-million-dollar formulation batches. ZDL valves guarantee process integrity at every critical juncture, maintaining absolute purity from the distillation unit down to the exact point of use in the formulation suite. Assessing your own facility's dead leg risks against the latest ASME BPE guidelines is a necessary step for modern compliance. In high-purity manufacturing, the zero dead leg valve is the foundational engineering standard that separates a vulnerable system from a scientifically unassailable one.